Hi students, my name is Niyati Said and thanks for watching Wikipedia word videos. My topic for the presentation is chromosomal compaction. So let's proceed towards our topic, that is chromosomal compaction. First of all, let's know in short, okay? As we know, if the DNA from all 46 chromosomes in a human cell nucleus was laid out end to end, it would measure approximately 2 meters. However, the diameter would be only 2 nanometer. Okay? Considering that the size of the typical human cell is about 10 micrometer. Okay, the DNA must be tightly packed to fit in the cell's nucleus. At the same time, it must also be readily accessible for the genes to be expressed. During some stages of the cell cycle, the long strands of DNA are condensed into compact chromosomes. There are a number of ways that chromosomes are compacted to fit in the cell's nucleus and be accessible for the gene expression. Okay? In the first level of compaction, stretches of DNA double helix wrap around a core of 8 histone proteins at regular intervals along the entire length of the chromosome. The DNA histone complex is called as chromatin. The bead-like histone DNA complex is known as nucleosome. Okay, this is nucleosome. This is the basic unit of compaction, and there are about 5 types of histone proteins: H1, H2A, H2B, H3 and H4. Okay, 4 of them are H2A, H2B, H3 and H4. They occur in pairs to produce histone octamer. Okay, 2 of H2OA and 2 of H2B, 2 of H3 and 2 of H4. That means 8. So they forms histone octamer called nucleosome body or the core of the nucleosome. Okay, their positively charged ends due to basic amino acids are towards the outside. They attract negatively charged strands of DNA, and about 166 base pairs of DNA is wrapped over nucleosome body for 1% turn to form nucleosome of size 110 into 60 angstrom. Okay, DNA connecting two adjacent nucleosomes is called intrabead or linker DNA. It bears H1 histone protein called plugging protein and that acts as a marker protein. Okay, length of linker DNA is varied, such as it is about 70 base pair long, and the nucleosome and the linear DNA together constitute chromatosome. Okay, nucleosome chain gives a bead on string appearance, as I have told you earlier, motiyo ki mala, under electron microscope. The beaded string is coiled to form cylindrical coil or solenoid having 6 nucleosome per turn. Okay, remember, 6 nucleosome per turn. Okay, actually the nucleosomal organization has approximately 10 nanometer thickness, which gets further condensed and coiled to produce a solenoid of 30 nanometer diameter. Okay, that means after nucleosome, the higher level of compaction is solenoid. Okay, this solenoid structure undergoes further coiling to produce a chromatin fiber of 30 to 80 nanometer and then a chromatin of 700 nanometer. Okay, as you can see, this is a double duplex DNA and nanometer in length, and then level of compaction goes and it formed nucleosome, which is about 11 nanometer. Okay, and then it got converted into a solenoid like structure, which is about 30 nanometer. Okay, and then ultimately it gets converted into a chromosome, which is mitotically active. Okay, and which is about 700 nanometer. Okay, Eukaryotic chromatin compaction, how it takes place? First, a DNA or the double DNA it's get converted into nucleosome. DNA is about 2 nanometer in diameter, and nucleosome is about 10 nanometer in diameter. Then it gets converted into solenoid-like structure, which is about 30 nanometer in diameter. And then it gets converted into chromatin fiber, which is about 30 to 80 nanometer in diameter. Okay, and then it gets converted into chromatid, which is about 700 nanometer diameter, and then finally it's converted into chromosome, which is about 1400 nanometer diameter, and which is mitotically very active. Okay, and so that it takes part in meiosis and meiosis. Okay. Eukaryotic DNA with a typical length of dozens of centimeters should be orderly packed to be readily accessible inside the micrometer-sized nucleus. Okay, if stressed end to end, a single set of human chromosome will be over 1 meter long, as I have told you. Okay, yet the cell's nucleus is about 2 to 4 millimeter in diameter. Therefore, the DNA must be tightly compacted to fit. That's why compaction takes place, to fit inside the nucleus. Okay, the compaction of linear DNA in eukaryotic chromosome, they involve interaction between DNA and various proteins. Proteins they bound to DNA are subject to change during the life of the cell. Okay, then comes the nucleosome. Okay, nucleosome, if we talk about the repeating structural unit within eukaryotic chromatin is a nucleosome. That means it is a basic structure. Okay, it is composed of double-stranded DNA, which is wrapped around an octamer of histone proteins. Which histone proteins? Those 4 histone proteins: H2A, H2B, H3, and H4. Okay. An octamer is composed of 2 copies each of 4 different histones, as I have told you, and at 146 base pairs of DNA, they make 1.65 negative superhelical turns around the octamer. Okay, as you can see, it are the octamers 1, 2, 3, 4, 5, 6, 7, 8. 8 histone molecules, they make up the octamer. Okay, and this is the nucleosome structure, and this is a linker histone protein. This is a DNA, and this is the H1 linker. Okay, which wraps around the octamer proteins. Okay, so nucleosome, you can say that subunit of chromatin, which is composed of a short length of DNA wrapped around a core of histone proteins. A human genome that contains about 3 billion nucleotide pairs, which are organized as 23 chromosome pairs. If uncoiled, the DNA contained by each of those chromosomes would measure between 1.7 and 8.5 centimeter long. This is too long to fit in a cell. Moreover, if the chromosome were composed of extended DNA, it is difficult to imagine how the DNA could be replicated and segregated into 2 daughter cells without breaking down, isn't? In fact, chromosomal DNA is packed into a compact structure with the help of the specialized proteins called histone proteins. These are the histone proteins. Okay, the complex DNA plus histone in a eukaryotic cell is known as chromatin. Okay, the fundamental packing unit is known as nucleosome. I'm again repeating, it is known as nucleosome. Each nucleosome is about 11 nanometer in diameter, and the DNA double helix wraps around the central core of the histone protein as an octamer, as you can see here, to form a single nucleosome. A second histone protein, which is known as H1, you can see as it is illustrated here, fastens the DNA to the nucleosome core. Okay, it fastens the DNA to the nucleosome core. The total mass of this complex is about 100,000 daltons. Okay, I repeat, the total mass of this complex is about 10,000 daltons. Okay, nucleosomes, they are usually packed together with the aid of a histone H1 to form a 30 nanometer large fiber. Okay, as a 30 nanometer fiber, the typical human chromosome would be about 0.1 centimeter in length and would span the nucleus 100 times. Okay. This suggests higher order of packaging to give a chromosome the compact structure seen in a typical karyotype or in the metaphase stage of the cell. Okay. This is the illustration. This is the linker region, that is H1, and that wraps around the octamer four types of protein, that is H2A, H2B, H3 and H4. Okay. The nucleosome with about 8 histone proteins plus 146 nucleotide base pairs of the DNA, and which is the core of the nucleosome, which is about 11 nanometer in diameter. Okay. So this is the diameter of the nucleosome, and this vary in length between 20 to 100 base pairs depending on the species and the cell type. Okay. And this structure gives us the appearance of the beads on a string, that is motiyon ki mala. Okay. This structure shortens the DNA length about 7-fold. Okay. Histone proteins are basic. They contain many positively charged amino acids such as lysine and arginine, as I have told you earlier, and these bind with the phosphates along the DNA backbone. These are the 5 types of the histone. H2A, H2B, H3 and H4 are the core histones, and 2 of each make up the octamer. H1 is the linker histone that binds to the linker DNA. It also binds to the nucleosome but not as tightly as core histones. Okay, this is types of histone proteins, okay, which form the core histones are the H3, H4, H2A and H2B, and this is the linker protein, that is H1. Molecular weights are written here, and now see the degree of conservation. Then H3 is highly conserved, H4 is also highly conserved, H2A is moderately variation among the tissues and the species is seen. Same is the case with the H2B. Okay. H1, it varies markedly among tissues and species. Okay, and H1 degree, that is not in your course, so I'm not explaining here. Okay. This is the nucleosome that shows the linker histone and the non-histone proteins. This is the histone octamer, and this is the linker H1 protein, or the H1 means this is the DNA. This blue part is a DNA, and these are the proteins. Okay, and these are the proteins, and these are the non-histone proteins. NHP, that means non-histone proteins. Okay, chromatins at different packaging level. 30 nanometer chromatin fiber, that means a condensed form, and the 10 nanometer chromatin fiber, that is a less condensed form like a thread of a bead. Okay, 30 nanometer, that means it makes a solenoid. It plays a role in the organization and the compaction of the chromosome. Nucleosomes join to form 30 nanometer fiber. What is that, students? That is a solenoid. Okay, nucleosome associate with each other to form a more compact zigzag structure fiber of 30 nanometers. Okay, histone, that is H1, it plays a role in this compaction. At moderate salt concentration, H1 is removed. The result is the classic beads on a string morphology, and at the low salt concentration, H1 remains bounded. Okay, beads associate together into a more compact morphology. Okay. The solenoid defines the packaging of DNA as a 30 nanometer fiber of chromatin and results from a helical winding of at least 5 nucleosome strands. Okay, in eukaryotic cells, 146 base pair of DNA are wrapped approximately 1.65 times around the histone octamer. Okay, which together are called as nucleosome. Histone H1, which is not a part of the binding histones, tightens the DNA bound to 8 protein complexes. The nucleosomes, which at this point resembles bead on a string, are further compacted into a helical shape via NH2 terminal protein interaction of the octameric histones called solenoid. Okay. DNA packed into solenoids, unlike DNA in nucleosome form, is not transcriptionally active. Okay, with more packing, solenoids are able to become increasingly more packed, forming chromosome. At this point, solenoids coil around each other to form loop. Okay, followed by rosette. Rosette, it consists of 6 connected loops. Okay, then a coil, and at last 2 chromatids. The end result is the metaphase chromosome. I repeat, the end result is the metaphase chromosome. The completely condensed chromatin has a diameter of about 700 nanometer. Okay. So that 13 nanometer fiber, it shortens the DNA length of the DNA molecule 7 folds. Its structure of 30 nanometer fiber has proven difficult to determine. The DNA conformation may be substantially altered when extracted from the living cells. And the two models have been proposed, that is solenoid and the three-dimensional zigzag model. Okay. This is a solenoid, which is about 30 nanometer in diameter, regular spiral configuration that contains 6 nucleosomes per ton, as I have told you. Okay, and this is the three-dimensional zigzag model. The contrary to the solenoid model, it is irregular in configuration, where nucleosomes have little face-to-face contact. Further compaction of the chromosome. So far, the DNA have been shorted about 50 folds. Okay, the third level of compaction involves interaction between 30 nanometer fiber and the nuclear matrix. Okay, the nuclear matrix is composed of two parts, that is nuclear lamina and the internal matrix proteins, with and 10 nanometer fiber and the associated proteins it form. Okay. The third mechanism of DNA compaction involves the formation of the radial loop domains. This is the radial loop domain illustration. This is a DNA that runs out and forms a loop kind of structures. Okay, and this is 30 nanometer fiber. Okay. This level of compaction involves a new proteinaceous structure called the chromosome scaffold. Okay, the scaffold itself is made up of several protein components that include the lamins and the topoisomerases. The lamins are primarily structural, while the topoisomerases are necessarily for relieving supercoiling tension that builds up in the double helix and ensure that a DNA molecule remains intact through the process of mitotic division. Okay. The 30 nanometer solenoid is anchored to a chromosome scaffold at discrete site called scaffold attachment region, that is SARS, and are organized into a radial loop-like domain of variable length. Okay, which is about 5000 to 20 base pairs. Okay, and this MAR is a matrix attachment regions. I hope it's clear to all of you now. Further compaction of the chromosome, the attachment of the radial loop to the nuclear matrix is important in two ways. That it plays a role in gene regulation. It serves to organize the chromosome within the nucleus, and each chromosome in the nucleus is located in a discrete and overlapping chromosome territory. Now come to the facts. I hope you would love to know about it. The length of a DNA in a single human cell is about 1.74 meter. Okay, and the range of the DNA length in human chromosome is about 1.4 centimeter to 7.3 centimeter. The human cell nucleus is only 5 microns in diameter, and chromosome at mitosis are 2 to 10 microns in length. The length of the genetic material is reduced about 7-fold by packaging the DNA around nucleosomes. Solenoidal winding of 10 nanometer nucleosome fibers into 13 nanometer nucleosomal arrays further compacts the genetic material around 5 to 6 times. Okay, I hope it's clear to all of you. So this comes to an end. In the next section, we'll be studying about the heterochromosomes and the euchromatin. Okay, so till then, stay tuned and keep watching Edupedia Word videos.